So welcome to chapter 15. I really like these last three chapters. 12, 13, and 14 were pretty nice, basic background of our genetics. But I think these last three chapters, maybe it's because so much of this work is what's currently going on, and so it's a little bit more exciting and interesting to me. But also chapter 15 is also kind of taking those concepts we've talked about in the last few chapters and turning them into what it means in the real world. And so I kind of like this chapter. So we're talking about the chromosomal basis of inheritance. So Mendel's hereditary factors were abstract when he proposed them. So he was just saying something is being passed from father to child, from mother to child, and then passing on to their children. But he didn't actually know what a chromosome was. He didn't know what DNA was. And it wasn't actually until like the 1950s that we actually agreed that it was chromosomes that were the hereditary factors. So that's what, 70 years ago? The people who actually figured this out, some of them are still alive. So this is pretty recent that we actually figured out what genes and chromosomes are. So today we can show that these specific factors, your hereditary factors, are genes, and that genes are located on chromosomes. And you can actually tag a particular gene, and you can see a particular gene on your chromosomes by tagging them with a fluorescent dye that will highlight that specific gene. So here is Fleming in 1882, Walter Fleming, drawing out what was going on in mitosis, what he was seeing in cells, and how he was able to see chromosomes moving during mitosis and moving to create those two cells. Here, this is tagging a gene. So these chromosomes are all red, except these yellow spots. And those yellow spots are, these are sister chromatids. So you have just multiplied your genes. So you have two copies of each allele, or you have two copies of each gene. And now there is sister chromatid. So you have 4 copies of that gene. So that's the 4 yellow dots that you're seeing. That is being tagged. And if you were to stop and sort out these genes by size, you'd be able to say this gene is on this particular chromosome. And if you wanted to tag out a different gene, you could create a tag for another gene. So cytologists are people who work on cells. Cytologists worked out the process of mitosis in 1875 and meiosis in the 1890s using improved techniques of microscopy. So here, being able to see chromosomes, this is obviously very good microscopy that Fleming was doing, and looking at these cells in these different stages. By the way, you should be able to tell me what stage of mitosis this is when those sister chromatids have been pulled apart. So just make yourself a note to yourself. There's a potential test question. Biologists began to see parallels between the behavior of Mendel's proposed hereditary factors and chromosomes, so we could see how chromosomes are being sorted out into different cells. Around 1902, 2 scientists, Sutton and Boveri, along with many other people in their lab, all lab work is done by a lot of people in the lab working together, but usually just the 2 people in charge are the ones who put their names on it. So Sutton and Boveri independently noted these parallels and began to develop the chromosome theory of inheritance. So how chromosomes could be passed down from parent to child and have that inheritance factors on it, how they could have the genes on it. The first solid evidence associating a specific gene with a specific chromosome came in the early 1900s from the work of Thomas Hunt Morgan. And so he did a lot of experiments, along with all the people in his lab, did a lot of experiments to show that chromosomes were the location of Mendel's heritable factors. In his work, he looked at this fruit fly called Drosophila melanogaster. Drosophila melanogasters are fruit flies, and they're very common. And because they're common, they also have other things that make them very easy to use as lab models. So for genetic studies, you want to see how genes are passing on to students, or how genes are passing on to their offspring. They have a lot of offspring, so that makes it easy to study. Instead of it's just, one child and flipping the coin that you have the gene show up or not. You have a lot of offspring, so you can see you can develop very good mathematical models for your genes. A generation can be bred every 2 weeks. So you are not waiting 25 years for a person to have a child and see if they pass on a disease. 2 weeks later, you have children. 4 weeks later, you have grandchildren. 6 weeks later, you have great-grandchildren. So very quickly, you can see a lot of generations. And they only have 4 pairs of chromosomes. So it's a little bit easier to tease out on those 4 chromosomes what is going on compared to if you had, some organisms have 50 or 80 or even over 100 pairs of chromosomes. Okay, Morgan noted the mutant phenotypes. He noted the wild, let's start this over again. Morgan noted the wild type phenotypes. Wild type phenotypes are what he would call normal, or what are most common. So in fruit flies, most of them have red eyes. So red eyes were called the wild type. So you'll often see WT listed by something as being the wild type. Alternatives were called mutants. So this fruit fly has a white eye, so it is a mutant. So the first mutant he discovered was a fly with white eyes instead of wild type red eyes. So if you are talking about a fly that is wild type, it would have red eyes. If you're talking about a fly that was mutant, it would have anything that's not red. So here it has white eyes. It does not mean that wild type is dominant. It does not mean that wild type is anything else. It just means that it was most common in that population he was looking at. So in one experiment, Morgan mated male flies with white eyes with female flies of red eyes. So he had white eyes that were mutant and red eyes that were the wild type. And the F2 generation all had red eyes. The F2 generation had a 3-to-1 red to white eye ratio. But only males had the white eyes. The females did not have white eyes. So from this, he reasoned that white-eyed mutant alleles had to be located somewhere on a sex chromosome, somewhere on the X chromosome, supporting the chromosome theory of inheritance. So those males, because they only had one X chromosome, if they had that white chromosome come up, if they had the white allele come up on that one X chromosome, they were going to have that illness. So they were going to have that mutation. But if you were a female, you had 2 chances of having the dominant phenotype come up. So, or if you were a female, you had 2 X chromosomes. You had 2 chances of having the dominant allele show up. So you were more likely to not have that eye color. So Morgan's discovery of a trait that was correlated with the sex of flies was very important to developing the chromosome theory of inheritance. So all mammals have 2 types of sex chromosomes. All mammals have a large X chromosome and a small Y chromosome. And if you end up with 2 X chromosomes, you are going to be genetically developed as a female, or typically you should genetically develop as a female. If you have an X chromosome and a Y chromosome, you will genetically develop into a male, typically. Well, aren't these pictures backwards? So your mom has 2 X chromosomes, dad has 2 X, or mom has 2 X, I'm sorry. Mom has 2 X chromosomes, dad has an X chromosome and a Y chromosome. That means dad's sperm, dad's gametes can have either X or Y chromosomes. Mom has 2 X chromosomes, so all of her eggs are going to have an X chromosome, half from, the 2 different types of the X chromosomes she has. That means that either you can have X from dad and X from Y. You can have X from dad or X from mom come together to make XX, or you can have Y from dad and X from mom, giving you an XY chromosome, giving you either a female or a male. That's not the only system that is used genetically, though. Some insects have a XO system. So if you have 1 X chromosome, you are going to develop as a male, and if you have 2 X chromosomes, you develop as a female. So these little symbols here, this typically refers to females, and this typically refers to males. Birds use a ZW system. So in birds, having a Z and a W chromosome makes female birds. So your hens you'll have hens, compared to 2 copies of the Z chromosome, which make you male. And some insects also use a haploid-diploid system. So if you are diploid, you develop as a female, and if you're haploid, you develop as a male. So the queen lays her eggs. The eggs that get fertilized turn into, the queen either lays fertilized eggs that are going to turn into all the female bees, or unfertilized eggs that can develop into the male worker bee, the male drones. Okay, so some differences in different chromosomal systems, how they work out to be different sexes. And then also, short segments at the ends of the Y chromosomes are homologous with the X, which allow the Y and X to behave like homologs during meiosis in males. So when you have homologous chromosomes lining up, that Y and the X can kind of line up together so that they're able to be sorted out appropriately during the process of meiosis. And genes on the Y chromosome are called SRY. This is for sex-determining region on the Y. So if you have a Y chromosome, then you should have this SRY gene that is responsible to develop testes in an embryo to go on and masculinize that embryo and turn that embryo into a male. So that's kind of the background. Then also another definition to know is a sex-linked gene. A sex-linked gene is a gene that is located on either sex chromosome. So any gene on your X chromosome, any gene on your Y chromosome is a sex-linked gene. If they are on the Y chromosome, they're called Y-linked genes, and if they're on the X chromosome, they are called X-linked genes. The Y chromosome is pretty small. It's only about 25 proteins on it, 78 genes that we've identified so far. The X chromosome has about 1,100 genes, though, and those aren't necessarily based on sexual development. They are just any gene that you need. So genes for being able to see colors are associated with the X chromosome. In cats, the fur color is on the X chromosome. So lots of just normal genes are on your X chromosome, not purely sex genes. Okay, so the X chromosome has many characters unrelated to sex. The Y-linked genes are typically related to sex determination. So either have the Y chromosome expressing those genes to masculinize the fetus, or don't have the Y-linked genes. They're not expressing those genes, so they are not going to masculinize the baby. The baby is born as a female. Okay. X-linked genes are going to have their own specific pattern of inheritance that's a little bit different than what we see in any of your autosomes. For a recessive X-linked trait to be expressed, a female needs 2 copies of the allele, right? If she has one normally functioning allele, then she's not going to express the recessive trait. A male only needs 1 copy of the allele because he only has 1 copy. So if he inherits an X chromosome that expresses any sort of recessive disease, he's going to have that disease. So a female needs to be homozygous. She needs 2 copies of the allele. A male only needs one copy of that X-linked trait because he only has 1 X chromosome. So he only needs 1 copy. He will be hemizygous. Because of this, X-linked recessive disorders are more common in males than in females. Okay? So by having 2 X chromosomes, females protect themselves from recessive disorders because females have 2 chances to have a normally functioning allele. Males only have 1 chance, and if they get a recessive disorder on their X chromosome, too bad they have that disease. Okay, so make sure that makes sense to you. So I want you to really stop and understand that because this is a pretty important concept for the rest of what we're talking about this semester. X-linked recessive disorders are more common in males, much more common in males than in females. Okay, some disorders caused by recessive alleles on the X chromosome in humans. There's X-linked color blindness, Duchenne Muscular Dystrophy, and hemophilia. So these are genes related to your X chromosomes. These are genes on your X chromosome. And because of that, they are more likely to be seen in men than in women. Men are much more likely to be colorblind Men are much more likely to have Duchenne muscular dystrophy, and they're much more likely to be a hemophiliac, because if they have that one recessive allele, that's the only X chromosome they have, that's the only allele they have, they're going to have the disease. Whereas females, if they get one recessive allele, they have another chance. If their other X chromosome is functioning normally, has that normal, the normal healthy allele, then they are not going to have the disease. Okay, and then let's also talk about X inactivation of female mammals. So here we have a calico cat. Calico cats have to have two X chromosomes to be a calico cat. So calico cats are almost always female. And that is because of this process called X inactivation. So in mammalian females, you have these two X chromosomes, and during embryonic development, one of them is going to be randomly inactivated so that you're not overproducing your X chromosome proteins. And so it's going to randomly inactivate and condense down. That condensed chromosome is called a Barr body. And so in most female cells, you will see a Barr body. Okay, and then if a female is heterozygous for a particular gene on the X chromosome, she will be a mosaic for that character. Mosaic means what you get like in a calico cat. So here she is heterozygous. She has one allele for orange fur and one allele for black fur. That fur color, both of them are on the X chromosomes. So in some cells, if the X chromosome that inactivates has the orange allele on it, those cells are only going to express black pigment. If the opposite happens, these cells have the black allele inactivated, they're only expressing the orange pigment. So she'll have spots where her fur is orange colored and spots where her fur is black colored, depending on which chromosome was inactivated, what Barr body she has in that cell, and which X chromosome is active. This only happens for your X chromosomes. If you have more than one X chromosome, this is all that happens there. You don't have Barr bodies based on any other chromosomes. These are only X chromosomes. Okay, so this is where we're going to pause at here. We'll come back and talk about linked genes in the second part of this lecture. So we've talked a lot about sex-linked genes, and we talked a little bit about the history of figuring out how chromosomes on DNA are what is hereditary matter. I'll see you back for part 2 of chapter 15. Bye.